is he? Oh, he's there.
All right, good morning, good afternoon, good evening. Welcome to our PolarConnect event. I'm Janet Warburton. I work with the Arctic Research Consortium of the United States, and we will be hosting this event today. I'm assisted with Judy Fonstock, who's in New Hampshire, and the research team and Lucy Coleman are standing by. If you haven't used Zoom before, you can switch the view to see the speaker or the gallery. The gallery is kind of a fun view because you can see all the little people's heads and classes and kind of get a sense of who is joining us today. So today's a really special day. It's December 1st, 2022. We're celebrating Antarctica Day. Today's presentation is focusing on what is Antarctica Day? Why do we celebrate it? And we'll also hear from our teacher, Lucy Coleman, who is down in Antarctica in a very special part of Antarctica. She'll describe that in a little bit. And she and the research team have been there for a while, and they've been working on microbial interactions in Antarctica lakes. And she'll introduce her team and her main researcher in a little bit. Before we turn it over to Lucy and the team to talk about Antarctica Day, there's a few things about Polar Trek and why you are all gathered here today. The first thing is, I gotta shift my slide. There we go. Whoops. Is that if you have just joined us and you know how to use the chat feature, please get into the chat box and open it up and introduce yourself. If you're with a school or a classroom, tell us where you're joining us from today. It's always nice to get a sense of who is participating. The program that Lucy is part of and that Judy and I work with is called Polar Trek. It's where we put teachers with researchers in the polar regions. We've been doing it since 2004. We're very happy to be supporting Lucy and her research team out of Miami University. And to date, we've had over 200 teachers that have traveled to the polar regions, both the Arctic and the Antarctic. Today, we hope you'll ask some questions. And as you ask your questions, one of the best places is to put them in the chat area. Judy will be trying to monitor that, and we will jump in during the presentation and ask questions if we need to. Otherwise, we'll hold them till the end, and Lucy and the team can respond to them. We will try to do some live questions. Sometimes that's challenging with a big group like this, but if we do live, you put up your, click on the little hand icon and we'll ask your question live. So with that, I am going to turn this over to Lucy to introduce herself and the team and get us started for Antarctica Day. Go ahead, Lucy.
All right, so welcome everybody, and thank you so much for joining us, and happy Antarctica Day. I'm here in the McMurdo Dry Valleys with Dr. Rachel Morgan-Kish, and we're at Lake Frixel, which is one of the lakes that we've been studying on our expedition. And I think I'm going to be daring and hope that our internet connection is good enough for us to just go outside here for a minute. We had some troubles getting connected and staying connected to the internet yesterday, but today seems pretty good. So I just want to show you around just to start off with. So hopefully you can see the screen. We were in our little hut here at Lake Frixel, but let me just show you what our view is here. So there's like a glare on the computer. Yeah, that's the lake. That's Lake Frixel. In the background, you can see the huge Canada Glacier. And let's see, dare I step away from the hut some more to give you an even better look here at the lake that we've been spending a lot of our time and energy on. Yeah, so it's actually December 2nd here. We're one day ahead of you. So we celebrated Antarctica Day yesterday, but we wanted to um, have the event on December 1st for you. So in the background here, you can see some of our labs. They look like little, I don't know, cubes. We've got a lab for different occasions. So there are 4 different labs depending on what kind of work we're doing. Um, and here in the background where Rachel is, hey Rachel, this is our main living hut, which is the building that I was just in. And yeah, so this is where we've been spending our time. You can see some fuel barrels back there, beautiful mountains, and it's a bright sunny day here. Um, and it's just kind of funny, the weather changes here pretty frequently. Right now, it's just like the best conditions we ever see here. Sunny, calm, and just beautiful, but it can be also pretty cold and windy here as well. So anyway, this is where we are. We've been here for about 2 weeks. I'm going to go back inside because it's chilly. It's about, I don't know, would you say it's like 20 degrees out or so? Yeah. And I don't think there's any wind chill right now because it's so calm. Yeah, but I'm excited that I could I could show you. It's just pretty wild that we're so far away from each other, and yet I can show you what it's like at this remote outpost. It feels like the middle of nowhere, but we're a lot more connected than you would think.
That was
Um,
amazing.
yeah.
Yeah,
Yeah.
I'm going to go back to sharing my screen, and you can keep talking while I get set up on my end, Lucy.
Yeah. Yeah, no worries. I see that somebody asked about the Starlink satellite. We're not on it. I don't know what our system is, but I think they're just testing Starlink at other locations, but it's not here yet. Okay, so I want to introduce my team. So go back one slide. So we have, as I mentioned, Dr. Rachel Morgan-Kish, and another PI is Brandon Briggs in the blue shirt there. And we've got some grad students. So from left to right, we've got Cody dressed in black. Rochelle has the plaid shirt on. Eckhart is our undergraduate, who is at Miami University with Rochelle. And then on the far right, we have Brad. So Brad and Rochelle and Eckhart are from Miami University in Ohio. And Cody and Brandon are from the University of Alaska. Yeah, so that's our team of 7. And we're here to celebrate Antarctica Day. So yeah, that's exciting to be the first live event from the Dry Valleys. It's amazing. This is like, yeah. Okay, so we're here to celebrate the Antarctic Treaty, which is an amazing testament to what we can do with international cooperation. So the Antarctic Treaty was signed on December 1st, I think in 1957 or 1959. And what happened was, Antarctica is kind of the last unexplored continent on the planet. And I think a lot of countries were starting to eye Antarctica for different purposes. And it was kind of a mashup of different countries having different claims to the territory. And there were people, I think, who could anticipate conflicts over that. I think there were some people that wanted to use the continent that they saw as a wasteland for things like nuclear testing. And so the international community came together to designate Antarctica as this place that could only be used for peaceful scientific purposes. And so with the original 12 signatories, there are a number of nations that have since signed on to the Antarctic Treaty. And it puts the continent under governance of the nations of the world. which means that you can come here and do scientific research, but you can't exploit the resources. So if you wanted to take rock samples to learn about them, that's great. But if you're trying to mine for coal or minerals or something like that, no go. The Antarctic Treaty also prevents nations from staking their claim to new territories. And it also gives any one nation the ability to go to another nation's research site and inspect it and make sure that everything is above board there. And what it means for us is that we can come to the Dry Valleys and find this beautiful and pretty pristine ecosystem that is protected and available for us to do research and investigate. And we'll share a little bit more about that as our presentation goes on here. Do you want to add anything else about the Antarctic Treaty?
Just that I guess it governs, how we do science too, because we have to be really careful to preserve the environment and these fragile ecosystems for future scientists too.
Yeah. So it's a pretty big deal if we accidentally leave something in the environment that shouldn't be there. So around here, that translates to every researcher having some horror story about getting something stuck in the lake and
getting it out
by hook or crook. Okay, next slide. So we just getting to Antarctica is a bit of a challenge. We fly to New Zealand first, and in New Zealand we go to a huge gear warehouse where we get all of our ECW or extreme cold weather gear. So that means big red parkas and big huge bunny boots and everything that we need to keep us safe in very cold conditions. We also had COVID tests in New Zealand to prevent COVID from making its way down here to the continent. And then we flew down here to Antarctica. So you can move on to the next slide. To get to Antarctica, you have to fly on a military transport. On our flight south, we flew on a C-17, which alone is a pretty big adventure. So it definitely feels like a military airplane. And it was kind of cool on our airplane. One of the passengers was the Prime Minister of New Zealand, which was, I just think, really pretty cool. We land on the ice. So the picture in the lower right is the runway. And we were transported to McMurdo Station, which is in the next slide. So McMurdo Station is the largest research station on the continent. It's an American station, but other people can go there too from other nations. And it's right on the edge of the Ross Sea. So you can kind of see on the bigger picture there of the continent. The closest jumping off place to get here is New Zealand. So that's why all of the traffic headed this way goes through New Zealand. Next slide. So McMurdo Station has all of your basic amenities. It has dorms. It has a big cafeteria to serve up to 1,000 people who live and work here. It has labs. It has a small hospital and a heliport where you can catch helicopter rides to nearby field research sites. Next slide. At McMurdo Station, we got ready for our field season. That took us a little over a week, and we did a lot of preparation for that. So we picked up all of our gear which is in the science taxi there in the middle of the picture. So tents, sleeping bags, equipment, drills. Yeah. We also went to a lot of training. Some of that was about like how to recognize hypothermia signs, how to operate some of the equipment that they were giving us, how to set up tents, how to do fancy knots, how to use the satellite phones and things like that. And at McMurdo Station, we also needed to get our lab equipment ready. So that meant sterilizing hundreds of sample bottles, making sure the machines that we were taking out into the field worked, filtering water to take with us. There was a lot of lab work to get ready for our field season. Next slide. So when we left McMurdo Station, we flew in helicopters to the Dry Valleys. So we traveled across McMurdo Sound into this pretty interesting, well, very interesting place. It's the only part of Antarctica that I know of that is not 100% ice covered. So most of Antarctica is covered by the continental ice sheet, except for this small area, which is one of the reasons why it's so unique. Next slide. And once we got to Lake Bonny, which was our first research site, we traded in our other means of transportations for ATVs. So so that was been pretty fun to zoom around on the ATVs. Okay, next slide. So our first research site was Lake Bonny, and Lake Bonny was pretty rustic. It had the James Way, which is this curved building here. I heard that it was left over from the Korean War, so I think it was used before it got here, and this is the last year for the James Way. It's going to be torn down and replaced with something else. But anyway, you can see that it's perched right on the edge of the lake, as is Lake Frixel Camp, which is in the lower right-hand corner, and that's where we are, where I showed you around a few minutes ago. In both cases, we're camped out in tents along the lakeside here, and it's just beautiful. But these do feel like remote outposts, and it's kind of fun to zoom around on the ATVs and get a good distance away, and you can just see it's just this tiny speck in this vast wilderness. Okay, next slide. So at our remote camp, we're camped out, we're sleeping in tents. We each have our own tent. A lot of people have asked if our tents have heaters in them. We are the heaters in our tents. So they are not heated. They do get pretty cold. And the beginning of our field season was very, very cold. And I think we all stayed warm enough, but it was a challenge. You know, a lot of us were taking hot water bottles to bed and scrounging for extra sleeping bags to keep warm. The hardest part for me is to get out of the sleeping bag in the morning and get dressed. Like you're laying in your nice warm sleeping bag next to this pile of clothes that are certainly below zero
And not clean.
and not clean. It's just not enticing at all. Yeah, so there's a good question here about human waste. So yeah, and how we handle that being so close to the lake. So for a number of reasons, including governance by the Antarctic Treaty about what you can and can't do and keeping the landscape pristine. In addition to that, the human waste is a pretty big issue here, because whatever you leave here in the Dry Valleys will be here for a really, really long time. So if we were to leave our waste products, you know, laying around, they would stay here for decades. And so we have to collect all of our waste. When we go to the bathroom, the liquids go into a big, like 60-gallon barrel, and our poop gets collected into buckets that are then sealed up and then shipped to California to be incinerated. So all of that has to come with us when we leave. That is along with all of our trash, all of our recycling, it all has to be taken out with us. So in the picture here, you can see that our way of getting water is to chip ice and then melt it inside the hut. There's a picture of the inside of the James Way there where we're packaging up some recycling in those containers. You can see in the background, it has a kitchen and refrigerator and living space. Okay, next question or next slide. All right. So this slide shows, excuse me. This slide shows Lake Bonny, and we zoom around on it on ATVs. Our research site where we were doing our work was a couple miles from Lake Bonny Camp. So we took the ATV to and from our field research site. Lake Bonny is a permanently ice covered lake. So the ice stays on the lake year round. In the summertime, a moat will melt out around the edge of the lake, but it was far too cold for the moat to be anything but solid while we were there. Next slide. So this slide shows a map of the Dry Valleys and highlights the two lakes in particular that our team was studying. So we've got Lake Bonny to the west, and Lake Bonny butts up against the huge Taylor Glacier, which I'll show you pictures of. Lake Frixel is closer to McMurdo Sound, closer to the ocean. And you can see there are some pretty big differences between the two lakes. Lake Bonny is deeper. It has saltier water, so it's more saline. It also is more oxic, which means it has more oxygen in it. And these, you know, in contrast to Lake Frixel, which is shallower, has more fresh water, and is also very, very productive. So the contrast between the two lakes makes it interesting for us to study both of these. There's a question here about how many people can be at Lake Frixel and Bonny at one time. We've had between 9 and 10 people at each of these sites, and that's a tight fit. I think oftentimes there's closer to 5 people, but I think there's also been as many as 15 or so, which is really crowded. Okay, on the next slide, we highlight some of the main features of the lake or the lakes. And do you want to talk about this slide?
Oh, sure.
Yeah.
Yeah, so we study, so the lakes are completely dominated by only microorganisms. So they basically have a very simple food web that's can't support any higher life forms like fish, or even maybe like some small invertebrates or something. Lucy mentioned that they have a permanent ice cover. So to get to our microbes, we have to go through 10 to 20 feet of permanent ice. There's minimal input, so that means there's a few streams influencing some of the lakes, and importantly, there's no outputs, there's no outlets either. And so the impact of no outputs is through climate change, the lakes are rising because the lake levels are rising because it's warmer some of the summers, and again, that's because there's no outputs. When we are here right now, it's 24-hour daytime. So the sun never goes down. And then in the wintertime, the life here in the dry valleys has to survive complete 24-hour darkness. The permanent ice cover means that unlike like normal lake processes, the lakes never ever mix. So normal lakes have mixing times, which are really important for a healthy ecosystem, and our lakes never mix. And so that's really interesting for microbiologists because there's all of these like layers like a cake, and the microbes living at the top of the lakes have very different conditions compared to microbes living deeper in the lakes. And as Lucy mentioned, it's the only place where it's ice-free, and these lakes, even though like in the wintertime it gets really cold and dark, the water in the lakes is liquid year round. And so it's harsh, but it's also a refuge for the life that we study here.
Yeah. So if we move on to the next slide, you'll see a graph that shows kind of the lake as you go deeper and deeper. And so as Rachel mentioned, it's really stratified. So each of the different layers is almost a little bit of its own habitat. So you have these kind of micro levels where the different species of microorganisms have kind of found their happy place. So for instance, in the graph that we're showing you here, you can pretty clearly see the level that the brown algae is happiest at. It has enough light at that point and enough nutrients that it's kind of hit its sweet spot in terms of where it likes to live. And the other organisms kind of find their happy place too. And because there are these little microenvironments, when we go to sample the water, we take samples from a bunch of different depths so that we can kind of see and learn more about who's living at that level and what genes they have to survive in those particular exact conditions. Yeah, there's a question here. Do we see any birds at these lakes? We did see a skua when I went out with Mike and Renee yesterday. A skua is it's kind of like an Antarctic pigeon. It's not a pigeon, but it's kind of a scavenger bird.
Seagull kind of.
Yeah. Occasionally people find dead penguins here, and also dead seals. We've seen a lot of dead seals. And what happens is they are trying to get to the ocean and they just get discombobulated and go the wrong way, and they just keep going until they die. And so it's pretty sad, but we can't do anything to save them. So we have to let that natural process go. But in terms of like charismatic megafauna, we've seen a lot of dead stuff.
I will say one funny story. The last season I was here at Lake Frixel, there was actually a skua that showed up every day, and we could see him out on the lake ice untying our experiment. And so it was interesting, but also frustrating. And I went out there many times to like retie the experiment.
Skuas are known for being thieves too. That's pretty funny. All right, next slide. All right, so among the microbes that we study are the protists, and so these are single-celled plants and animals that are very, very small. And they are some of the key life forms that live in these lakes. So, and they form a critical component of the food chains here. And a lot of the questions that we have been focusing on for our research involve the roles that they play in the ecosystem. So how are they interacting with other members of the ecosystem? Sometimes they consume each other, sometimes they compete against each other, sometimes they're trading resources with one another. and they can be friends one day and enemies the next. And in some ways, they kind of keep each other alive and depend on one another. And we're really interested in understanding these interactions. And I kind of think of microbiology as being one of the big frontiers in science. We know a lot about microbes, but there's a lot that we don't know, especially when it comes to their role in geochemical cycling and the balance of carbon and sulfur in other ecosystems as well. So because this ecosystem is so isolated and there's only microbes here, we can learn a lot about them. Whereas in an ecosystem like the ocean, there are so many inputs and outputs that it's harder to isolate the role of the microbes in these geochemical cycles. Whereas here we can do fairly controlled experiments to learn more about these relationships. Would you say anything more about that?
Yeah, it's just that people have called the Dry Valley Lakes natural laboratories. So we can do manipulated experiments, but basically the lakes actually act as natural laboratories, and they're perfect places to study these organisms like the protists because again, the whole food web is dominated by microbes, and the protists are the plants at the base of the food web. So they're single-celled plants, but they're also basically the lions or the top apex predators of the ecosystems too. So they're also these basically work as single-celled animals. So they basically hunt other microbes. So that's another reason for people who are interested in these organisms, it's a perfect place to study them.
Yeah. Okay, next slide. Okay, so our major goals for this project, just to kind of sum up what we were just saying, is to understand how these microbes are interacting with each other, to identify which factors in the environment are influencing their interactions. So for instance, if they have access to more iron or more salt, how does that influence their interactions? And then a lot of our samples are being taken back to labs at the University of Alaska and Miami University, where the genes involved will be analyzed using technology that we don't have here out in the field. But understanding the genes can help us get a bit bigger picture of how they're interacting. Okay, next slide. Okay, so then that kind of leads to the next question of how do we do all of this research, because these microbes are fairly inaccessible. They're hiding under all of this ice, isolated, and they don't make it easy to to access. First of all, we have to get here to Antarctica and then to the dry valleys, and then we have to get to the microbes themselves, which is an adventure. So we use a couple of tools in particular for that. One is the Jiffy drill. And the drill, you can see the drill bit that we pulled out of the lake there. The drill has to be as long as the ice is thick. And so we keep adding sections of drill as we get deeper and deeper into the ice until we reach the bottom of the ice and water comes up through the drill hole, and we know that we've made a hole all the way through the ice cover. Some of our drills are 5 inches around and some are 10 inches around. And yeah, the ice is really thick. We've found that it's between 3 meters thick, and our deepest hole through the ice was closer to 6 meters. So that was a lot of drill flights or sections to get through. It was a huge challenge. Now, in some cases, depending on what we want to put into the lake to collect samples, we need a wider hole. So on the left-hand side here is a hole melter. It's a machine called a HOTC. It's connected to a generator, and we lower a coil into a drill hole, and the hot sea will melt the hole to make it wider, which in some cases is really helpful. So that's how we get through all of that ice. And then in the next slide, I'll show you here what we do once we have a drill hole open. So this is where we collect, or how we collect samples. So in the upper right-hand corner there, you can see a picture of Rachel and I don't know who that is,
That's Brandon.
with a winch. The winch lowers a device into the hole that will collect water. But one of the challenges we've had is that it's been so cold that and the lake under the ice is above freezing, which allows it to stay liquid. But as soon as you bring that water up into the cold air, it freezes. And so it's been so cold that it has been freezing really fast. So we lower equipment into the lake, we bring it up, and like 5 seconds later, it's frozen and like locked, and we can't do anything with it. So we've actually, for a lot of our sampling, we had to set up a Scott tent to protect our equipment and our own selves from the cold. So we were sampling inside the Scott tent. There's a question here about whether there are holes from years of past collections. The holes freeze. So within, depending on how cold it is, you can go back an hour later and there's already a crust of ice over the hole. So there's no hole that would last from year to year. However, we do, for some teams that work here, we do keep record of the GPS location of a hole so that we can go back to that same hole the following year and do multi-year collections at the same place.
And it's also for environmental records too. We have to record any activities or any interactions basically with the ecosystem. So that's the other reason we record all our activities on the ice. To do with the treaty.
Okay, next slide. Okay, so one of the tools we use for collecting is the Niskin, which is the device Brad is holding there. It slams shut at both ends when we get to the depth of water that we want to collect. And then we can analyze the data from different levels of water and collect samples from those depths. Next slide. Once we have the samples, we bring them back to the labs at the camps, and we do different procedures to preserve the samples. Sometimes that means flash freezing them. Sometimes we filter the water and collect the chlorophyll and nutrients from the water. And this is mostly for taking all of those samples back to labs in the rest of the world for further processing. Next slide. Okay, so along the way, I just wanted to share a little bit some of our challenges. So all of this that I've told you is kind of like what happens when things go well, but sometimes they don't go well. We've had a lot of opportunities for stuff like drills to get stuck in the lake. And, when it's so cold, things break a lot easier and we end up having to jerry rig them or find workarounds. And the right-hand side shows Rachel wearing her broken Niskin. And it's hard because sometimes we do all this work to open up a drill hole and set up the Scott tent and do all the things only to find out that like the equipment we need for the last step is broken. So there's a lot of troubleshooting that goes on down here too. And it's been a colorful experience. All right, so that's how our field season has been going. We're almost done here at Lake Frixel, and we'll head back to McMurdo Station tomorrow. We'll do some more sample processing while we're there and get things packed up and ready to be shipped back to the United States for us to do more work at the universities. Yeah, so let's have some time for questions here. What are people wondering about here?
Yeah, great presentation. Thank you. Lots of fun things that I hadn't heard about. So it is time to ask your questions. If you want to ask them live, you're welcome to do so. Just raise your hand or type them in the chat box. Where are the other team members?
So yeah, that's a good question. Some of them are lurking in the background. They're looking at us now. And some of them are not awake or up yet. So some of them are still in their tents, probably contemplating that big pile of cold clothes laying next to them.
It's also
well, what time? Yeah, I was going to say, what time is it there, since you mentioned time?
Yeah, it's 7:40
in the morning.
Morning. Yeah.
So they're taking a little bit of a break too after a long season. So it's our last day, so it's a little bit of a break. Yeah.
All right. Bill says, my students are wondering about what you do for recreation.
Sample the lake.
Yeah. It is pretty fun. We've had some really long days, but we've also had a chance to watch movies that we brought down on our devices. We read books. Cody brought a bunch of coloring books for us. So sometimes we just sit at the kitchen table and color.
Baking. There's lots of bakers here that we can bake cookies and other kinds of treats. We celebrated Thanksgiving. We hiked over to another camp called Lake Hoare, and we had a really great Thanksgiving dinner there and a really nice hike around the Canada Glacier.
Yeah, that does sound good. Julie wants to know, what's the reason for salinity differences in the lakes?
Oh, that's a good question. I just learned this the other day from Brandon. So this whole lake system was once connected to the ocean, and has and when the water levels receded, it became unconnected from the ocean, but that ocean water was filling up these basins. But the ocean, or the water over time, has evaporated, but the salt doesn't. So it's just become more and more salty. The other thing is, at Lake Bonny, there's Blood Falls, which is a super weird feature that I highlighted in one of the journal posts, but it has a lot of brine in it. So very, very salty water as an additional source of salt for Lake Bonny.
Okay.
The coldest.
Yep, go ahead.
Yeah, these other questions here. Is it easy to maintain the equipment? I would say generally no.
So we have a huge level of like non-science support staff in the station, and they basically maintain the equipment for us so that it's working really well. And then it comes to the Dry Valleys, and Antarctica is really hard on equipment. So there's often equipment issues, and it's good to have some level of knowledge about fixing equipment, or having good friends here that know how to fix equipment. Or lend you their equipment.
Like our drill was having some huge problems the other day, and within a half an hour we had somebody from our team here know how to adjust it. And we also got a second drill from another camp right away. So there's options when things go wrong.
I was gonna. Oh yeah. Go ahead. I was going to go
Sorry.
backwards a little bit because there was some you skipped to, but that's
Yeah.
okay. So from your class, actually, the second and third periods, they want to know what genes make different microbes stand out from each other from Madison.
Yeah. You want to go for that one?
Yeah, so the really obvious ones are microbes have different jobs, so certain microbes have genes for photosynthesis, right? Other microbes have different kinds of genes to use different carbon sources. So there's tons of bacteria that use different kinds of carbon. And then I talked about, some microbes can actually digest other microbes. So they actually have like these sort of microscopic digestive systems. But the other things that stand out are my lab actually has isolates of Antarctic algae especially, so we can do more directed studies on those. And we've discovered that there's a lot of special genes around photosynthesis that allow these organisms to survive permanent low temperatures all the time. One example are these proteins called ice binding proteins that basically bind ice and protect the organism from being injured from physical ice crystals, basically. They also have special genes actually to deal with high salt, because salt is a huge stress. And especially lakes like Lake Bonny have very high salt stress. So those are some different examples of the special genes that the microbes here have.
Cool. Another question from Miss Lively's class, period 3. How long do you spend collecting drinking water each day?
So usually we get a whole bunch at once and then don't have to collect water for a couple of days. Here at Lake Frixel, we can collect what we call glacier berries. So we take the ATV over to Canada Glacier, and there are these chunks of ice that have fallen off the glaciers, and we can take those big chunks of ice, we bring them back here and melt them. And when we do a big glacier berry collection, it can last a camp of 10 for maybe 2 or 3 days. And then we have to go back out and collect glacier berries again.
Wow.
But I think it also, yeah, it also highlights how much work we have to put into just survive here.
Yeah.
So there's our science, but then there's just a lot of activities like making sure we have drinking water, which take a lot of our time too.
Yeah.
Yeah, and a lot of resources like fuel for heating up or propane. Your second and third period class is wondering, is there rovers?
Rovers. Yeah, and I see this other question about robots, and that's my third period is my robotics class.
Oh.
I like
Cool.
Yeah. So there are a lot of autonomous electronics that are used here. And so, there are other science teams that use things like underwater robots. But here in the Dry Valleys, more commonly, there are not exactly robots, but there are a lot of electronics that are set up to record data autonomously. So for instance, we just put a big piece of equipment in the lake that will sample water year round. So every, I don't know, 2 or 3 weeks, the thing opens up, collects a sample, and then rotates around and gets ready to collect the next sample 3 weeks later. And so we need those kinds of equipments that can do that year round, because we can't stay here year round. It's just not a realistic proposition. So there are a number of stations around the Dry Valleys that collect data autonomously like that.
And there
Cool.
was actually an underwater ROV that was deployed several years ago in Lake Bonny that actually drove around in the lake and looked at blood falls like in the underwater, and Yeah, and that kind of NASA supports that kind of work. They're very interested in, deploying equipment safely in a harsh place. So,
Yeah.
and are also interested in the dry valleys as analogs of what life might look like on other planets.
Well, that kind of segues into this question from Elaine Hood. Can you please explain again why you're doing research there instead of, say, the US? Why Antarctica?
That's a really good question because it's a huge amount of effort to get here and live here and do this work with microbes. And maybe I'll take a crack at this and you can answer it too. But the thing is, these ecosystems are very unique. And as Rachel and I have both mentioned, these microbes are living in this very isolated ecosystem that's really controlled. And so in other parts of the world, like the ocean, you have so many inputs from so many different rivers and streams and sources of pollution and weather patterns that you can't really isolate what the microbes alone are doing nearly as easily as you can here, where they dominate the ecosystem and the ecosystem is pretty cut off from any of those inputs. And so to better understand those geochemical cycles, having this really controlled environment is super helpful. And then that helps us understand things like their role in the carbon cycle, their role in things like climate change. But what would you say about that?
Yeah, I can give two other examples of reasons why we do research here. So the first is that I mentioned the ecosystem is fragile, and that means that the organisms that live here are kind of canaries in a coal mine for having an extreme response to even moderate changes in the climate. So they're highly responsive to small changes in global climate. And I know there is a question that asked about evidence of global warming, so we could talk about that too. And then the microbes here, as I mentioned, have really specialized ways of dealing with environmental extremes. So they're also novel genetic resources for, dealing with environmental conditions that other life cannot even survive under. So for example, we have a project that basically we're using algae from here as models for plant stress, so looking for novel ways for plants to deal with environmental stress, which is supported by the Department of Energy, actually.
Okay, since you're talking about that, let's see, there was the question about, has Antarctica been affected by global warming and how does that impact your research? And there's a related question below also about climate change impacting your research efforts.
Yeah, so as Rachel mentioned earlier, the lake levels have risen, and so that here in the dry valleys, that brings up questions about whether this is a very stable ecosystem, which is how it's been for what we think are 1,000 years or more. So up until recently, the lakes have been pretty stable. There have been very few inputs, no outputs. But recently, with climate change, the glaciers are receding a little bit more, dumping more water into these lake basins, raising the level of the lake. So how is that changing this ecosystem? It brings up a lot of questions about stability and change and what that means for microbes, as Rachel was just saying, like how does that change things, even just those small inputs. On a larger scale, I would say also that Antarctica is being heavily researched right now to find out how stable the glaciers are. In some places on the continent, they're showing a lot of signs of flowing very quickly into the oceans. as compared to here in the dry valleys, the glaciers are relatively stable. So, there's a huge amount of ice in Antarctica, and if it were to go into the ocean, it would be a really big problem for the rest of the world. Yeah. Did you add anything to that?
I think just to point out that, a couple of things. So there's a big impact on the humans too. So as Lucy mentioned, our camps are really lakeside now, and that's because of the lake level rise, so actually Lake Bonny in particular has a very steep lake basin, and there's been I think several meters, like up to 15 meters of lake level rise, and they've actually had to remove lab buildings, which would be underwater now. And then the other thing is, our project now is a 3-year project, and we have 1 field season. So it's kind of hard for us on our project to sort of observe climate change, right? Because we're just here for 1 season. But we work with another project called the Long-Term Ecological Research Program, or LTER. And these LTERs are located all over North America, as well as several in the polar Arctic and Antarctic stations or ecosystems. And the McMurdo LTER has been here for more than 30 years. And so that's a really important resource for, observing climate change over a long-term basis. And so this long-term research is really necessary to track changes in these ecosystems over several decades. So we're really lucky that we have support for these long-term projects as well.
All right. And I was just typing in the chat that related to the LTR team, we do have 2, we have 1 teacher, Bill Hensky. I think his class was joined earlier. He will be headed to Antarctica with the LTR team. And we have another educator, Elaine Krebs, who will be headed to the South Pole Station. So we have lots more about Antarctica coming up this month and in January. So looking forward to hearing about their experience and their reports. A few more questions before we kind of wrap up here. I think it was Mrs. Lively's class, period 3. They wanted to know, what is the coldest it has gotten there? Maybe while you've been there, and then kind of if you know what the temperature is like during the winter, that would be interesting.
I'd have to look at the data on the winter, but the coldest it's been while we've been here, I think with the wind chill, the number that's sticking in my mind is -25. But that's with the wind chill.
I would say, I think it was -27.
Geez.
Yeah.
It was
That's pretty cold.
It was super
Yes. That's cold.
Record in Antarctica, not in the dry valleys, but at another location, I think is almost -80 in the winter time. But I think the record in the dry valleys is -50 F in the winter time.
Oh, goodness. All right. Let's see. There was another question. There's a lot of questions about food. I forgot what the one was earlier, but I think it was about what do you eat for meals? And then your class there, 2nd, 3rd period is how often is the pantry restocked? And what happens if you run out of food? So food, they saw the food in your background and are curious about it all.
And I see a couple of other questions about general living here. So yeah, our pantry gets restocked about once every week or 2, and we get a boatload of food or helicopter load of food from McMurdo. About that often. It includes a lot of frozen foods like frozen chicken and frozen steak and shrimp and stuff like that, and then canned food and packaged food and not very many freshies. Yeah, most of it is expired. Most of it is still pretty good, but every now and then we have to throw something out, and yeah. Let's see, we're not going to run out of food. We have so much food, so that's not really, yeah.
Good.
And there's some questions about sleeping and the 24 hours of daylight, and sometimes that throws us off. I think, I don't know about my teammates, but I think they have the same problem in that we go to bed, we pull the sleeping bag or beanie over our eyes so that it's dark in our minds anyway. But then in the middle of the night, I wake up and I jerk awake thinking it's broad daylight. I'm late for something. It must be like 8 in. And then I look at my phone and it's only like 1 in, but it's broad daylight. And I'll be like, oh my gosh, like I get all amped to get up and go do something when really it's still the middle of the night. Yeah. There's a question about sleeping in tents, like why do we sleep in tents instead of in the buildings? And the thing is that our building here, like this is our living space, and it's got the kitchen back there where Tina's having her coffee, and we've got a couch over here. There's no room in here to sleep, and so we sleep in tents, which is just fine, and that allows for each of us to have our own little refuge at the end of the day. Yeah, but it's rustic.
And it's the only opportunity for privacy.
Yeah.
So people appreciate their personal tents.
Yeah.
Great questions. Well, we're going to kind of wrap up here. I'm going to go back to sharing my screen real quickly. And if you didn't get your question answered, please feel free to post it to Lucy's journal, and she'll be happy to answer them. This event was recorded, and we'll also be sending out the link to everybody that registered, and we will post it online. Kind of some wrap-up slides before we close down here. This event was brought to you by ARCIS, and we want to make sure that you all know how to access the ARCIS website at arcis.org, and that we have a lot of materials there about the work that we do, and calendars and meetings that we host and publications. So be sure to check out ARCIS and become a member of that as well. We also want to give thanks to the National Science Foundation for funding Polar Trek over the years, and to all the staff that have worked with this as well. And a big thank you to Miami University and to Rachel in particular for bringing Lucy with you to Antarctica. We've been enjoying working with Lucy again. She was Polar Trek alumni, and it's been great to hear her second adventure to Antarctica and reconnect with the science that's happening down there. Thank you for a great presentation about Antarctica Day. It was great. And I'm sure you're looking forward to some different kinds of sights, food, and probably mostly a shower and laundry, clean clothes. With that, I'll say thank you again, and feel free to I'll let you close out there, Lucy.
Yeah, the shower is top of the list there. Thank you so much to all of the people in our communities that have joined us here today. I really appreciate you taking the time out of your day to learn about Antarctica Day and our field research here. I look forward to seeing all of you at home when we're back in the United States, and just very glad that you could join us here. So thank you. Thank you so much, especially to the teachers out there who have devoted your time to us today to sharing our project. Yeah.